let's talk about homology modeling and specifically how HHpred works. One moment. Let's plug in the good microphone. Ooh, is that better? I hope so. All right, so this is the basic schematic of HHpred. This comes from a very nice online slide set from Fabian Glazer, and yeah, really thanks to Fabian. Okay, so how does this work? Fine. HHpred is a tool that's going to give us a very good assignment of protein shape. And by accurately reporting the shape of a totally unknown protein, it's going to give us a pretty good idea of what that protein does, because shape is correlated to function most of the time. So yeah, HHpred ends up being an invaluable tool for investigating new proteins, how they work, what they're doing. So let's look. All right, so you start out, you make a target sequence alignment is the best way to do it. It can do it on one sequence, but if you put in several sequences, it goes much better, yeah. So if you're finding not just one new thing, but a group of related new things, that gives you a much more powerful HHpred result, basically. Then HHpred has a little bit of BLAST inside. So it has a thing called PSI BLAST, which is position specific iterated BLAST. So position specific means for each amino acid in your protein, yeah, let's say the first amino acid, methionine, what can that change to? So if it's always the first amino acid, it changes to nothing, and so there's nothing. But if you look at the second amino acid, let's say that's alanine, there'll be a certain percentage chance that it could change to anything else. So let's say your second is an alanine, so here's an example up here. There, you would make an alignment and say, oh, okay, occasionally it changes to arginine, never changes to aspartic acid, that's what D is, arginine is R. Really commonly changed to asparagine, which is N here, and so on. And so you build this thing, which is a position-specific matrix, a PSSM, for every protein at the start. And the PSSM plus the actual structure of your protein is going to be this thing that's called a profile. And HHpred matches profiles. Yeah. So it's going well beyond just trying to line up, one amino acid with another, which is like what BLAST was doing. But as you can see, it's got a little bit of BLAST inside. So you'll send in your sequences. HHpred will then go out and it'll say, maybe I can get you some more help. And it will find anything that is close enough to come up as a hit on PSI BLAST. And so it'll do this over and over. It's an iterated BLAST where it sort of refines the hits that you get and refines the statistics on those hits. Okay, so it'll get some more, and then based on your sequence and all these other sequences, it'll make this position-specific matrix. So if the thing you're looking for has no other sequences that are anything like it, then you really don't have a position-specific matrix, and you're not going to get great results from HHpred, at least from the sample end. You may still be able to pull something out. Then it's going to do an alignment based on this this profile. So it's not just aligning that you have an A here. It's that there's an A here, but also sometimes there's an N. Occasionally there's a G. There's never a D. Like that. It's a sort of a multivariate analysis, and it's able to do alignments based on this. So it's basically aligning the important stuff that doesn't change as much during evolution. And it's not relying on you or me to know what is important. It is just pulling these things out and saying, well, if it doesn't change, let's give that more weight, basically. Which is really powerful and is pretty much the best you can do. Okay, and so it'll generate position-specific gap penalties and probabilities of change for changing to each of the 20 amino acids. And if it's not represented, it'll give it some, generic, fairly big penalty, just because if you've never seen it in nature, we would not at least expect to see that going forward. And then it'll do a thing called an HH search, which is a hidden Markov model alignment. And so it's going to align your little profile that it's made. It'll predict the structure. It will align all the sequences it can find. It'll find the common points that change very little, and it'll make a lovely alignment. And then it will align that profile to other profiles that it has already pre-computed. So the other nice thing about HHpred is that they went through the databases of conserved structures and all the structures that have been solved, where we actually know the actual structure of the protein, and they computed these little profiles for all of those. And so then it'll find you the best match between your profile and the profiles that are in these databases. And you just select the databases to scan, and it works remarkably well. And the beautiful thing is, it is very fast, very, very fast. And yeah, just like the little green box over here says, easy to read results, including e-values and probabilities. It gives you the secondary structure, even if protein secondary structure, even if you didn't know that. It gives you a consensus sequence for, if yours is a little different from the norm for these, it'll tell you what the sort of average sequence that looks like this is. Yeah. So that's pretty good with position-specific reliability. Yeah, it's pretty much the best. Yeah, there are things that can do slightly better at each one of the things that HHpred does, but there's nothing that even gets close in terms of the amount of time it takes and being able to do all the things that HHpred does. And so this HHpred first came out in 2005, and it has only grown in use and popularity since. It is amazing. Yeah, my favorite bioinformatics tool by far. So, all right. What's the problem they were trying to solve? Well, the problem they're trying to solve is that if you have a new sequence, you can predict the structure that that protein will form if you can match it to an existing protein. And that's the truth of biology. There are only a few structures that proteins have ever been able to form that exist in the sort of catalog of life. And all these different things that we think of as, totally different life forms are mostly using a, frankly, copied, very basic set of genes that make proteins with certain shapes over and over and over again. Just little tweaks on that. That's what evolution gets you. So the key problem here is taking an unknown sequence and figuring out what protein should I model it based on. Because we've got good modeling software that can model based on, anything, as long as you can find the right thing that lines up to make a sensible model. Yeah. So that was the idea. So let's see. All right. So these modeling programs, which we will get to later on in the course, they can make you a model of anything, and the model will only be as good as the template that you chose. So they're all very dependent on template. There are some bits of modeling software like AlphaFold that will attempt a completely what we call de novo. That's DENOVO, like fold or ab initio, like from nothing. From the beginning, they will compute the fold. Those sometimes work, and if we can solve that, that's going to be a great thing. They're in progress. They do well, but they're not, they're not there. They're not reliably perfect. So it's still very dependent on what you're aligning in order for the model quality that you get out at the end. All right. Template search methods, like BLAST, will usually produce a non-optimal alignment, just because they're going from a little bit, remember that conserved word, and they're just trying to expand out. So they can't take an overall view of a protein and which parts are and aren't conserved. They just blindly will make a match, try and make it bigger, and then extend that match as far as they can, as far as the stats allow them to. Yeah. So yeah, it produces problems. There are better ways to solve some of these alignment problems. When you have more than 30% amino acid sequence identity, and remember, we're not saying 30% homology because they either are or aren't homologous. We're saying amino acid identity, which is 30% of the amino acids are identical between your thing and something else. When you got that, then automatic methods for sequence alignment are usually pretty good. Things like Clustal Omega will do a pretty decent job. When you are below 30% sequence identity, then manual alignment curation is required. So you'll use a computer program to generate an alignment, and then you will edit the alignment based on things that you know. Like you know that there's a particular active site. So we'll line up the amino acids that make the active site, because that's the thing that makes the enzyme go. You can predict the structure of your protein and say, okay, all these are in a helix and all those are in a helix, so let's align those, like that. There are a lot of ways to do this. Every misalignment you make is critical and will really mess up your model. So 4 angstrom deviation is a lot. It's 4 atomic widths basically out of whack that your alignment will be, or rather your model will be. Okay, so along comes HHpred. Great little program. This is from some of the benchmarking that was done around HHpred, and this does not appear in the HHpred original paper, this 05 paper, which is a good read. It's actually pretty simple. And you can see the old user interface that they used to have. It's kind of clunky and neat. It looks like 2005. So these are the various things they were working on. They tried a couple different versions of HHpred. And the fastest one that gave the best results was this one, this HHpred 2. So that's the one they ended up using. And so that's the one that we call HHpred now. 1 was fast but inaccurate, and 3 was a little slower and also less accurate. So basically higher bars are being able to match up more things that are definitely the same. There are methods that can do better. Like here's the Zhang paper. This one led to a different server, but see the difference there? This one takes 35 hours to do what HHpred can do in 15 minutes, and it only gets you, I don't know, a little less than 20% additional accuracy, additional homologs that it can pick up. So it can do better, but it's not going to save the day. And both these servers started at the same time. The Zhang server is now dead. Nobody maintained it. It has gone away. So you cannot use it. The program is still there. You could compile it yourself and run it at your leisure, and it might do a little bit better job. Also, the Zhang algorithm is only for fold recognition, so it doesn't do some of the other things that HHpred does. And so it's a limited tool that takes a whole lot longer to run. Yeah, HHpred all the way for me. Eventually, I guess somebody will do better, but this is where we are right now. Okay, what does HHpred do? So it's really good at remote homology detection. So remember, homology is only yes or no, but homology can be really close and obvious with amazing stats, or homology can be kind of distant and less obvious with not as good, amino acid identity, but still homologous. And HHpred is really good at finding the distant ones, finding the ones that other programs will not be able to find. It's as easy to use as BLAST. It's got a big box in the top. You paste something in and it runs, and it's done in a minute or two. Really good. And it'll take either one query or you can actually put in a multiple sequence alignment. Now to do that, and this is relevant for assignment 4, to do the multiple sequence alignment, you have to output the alignment in Pearson FASTA format. That's the correct format that HHpred is able to use, and a lot of other programs. Mr. Bayes, a bunch of programs. They're all happy with an alignment in that format. All right, so yeah. And then it does have a modeler attached to it. I don't think it's the best one, but it will model based on the HHpred results, and because it gets better results, you actually end up with a pretty good model. Yeah, it's not as fast as the ones we're going to use, but you can use it, you can try it, you can compare it. In fact, I may modify the assignment so that we do that. That seems like a good thing to do. Okay, so HHpred is not doing local alignment, and it's not doing global alignment, it's doing profile alignment. So it's a global alignment, but one sequence plus all the other versions of that sequence, all the other homologs of that can be identified, and it's aligning that and comparing it to an alignment of other things where it's a domain and we have a name for it. Yeah, so that's what these profiles are. And the profiles are going to contain these position-specific matrices, and they're also going to contain protein structure information. Some of this information may be predicted structure if we don't have the real structure, and some will be actual structure from the solved NMR or crystal structures, or EM structures for that matter. Yeah, and that also works really well. And yeah, they are superior to sequence-based methods by far. Okay, what's it looking at? It's looking at a position-specific scoring matrix. That's like the thing over here that we mentioned earlier. SciBlast will generate a position-specific scoring matrix once it finds various hits, and it refines this sort of PSSM, which is its little sort of model, its little matrix for your protein. The idea is that something like a glycine, a G changed to a lysine. Sometimes that may be common, sometimes it may not. When we take something like the Blossom 62 matrix, we just say, oh, glycine, alanine, yeah, or arginine +2 or -5 or something like that. And it's always the same. But of course, it might be totally tolerated in some parts of the protein and not at all tolerated in other parts of the protein. And if you have multiple examples of that protein that are all functional, that work in the world, then you can actually figure out which of these things are allowed to change and what they're allowed to change to. So it's pretty darn good. It's, yeah, a very nice way of doing it. So, yeah. Any one substitution can be different for different alignment positions. Yeah, in contrast to the way we do it with BLAST, with these PAM and BLOSSOM matrices. There's an example of a position-specific scoring matrix. And so each time you run one of these, it'll generate a brand new matrix just for your protein. There are ways to go in and, I believe, look at that matrix, see all the data from it in either SciBlast or in HHpred. I think you can unpack that. Takes a little more work, but yeah, it exists. Doesn't look as pretty as this. This is very nicely formatted with the colors and everything, but yeah, it's saying the same thing. Okay, so the name of the game is profile alignment, or profile comparison, and so you've got one profile and you compare it to another profile, and that's great. By the way, looking at this profile, can you tell what kind of protein this is? If you can figure it out based on this and tell me why, that's going to be worth an extra 10 points of extra credit. First 10 correct answers will get 10 points extra credit. So there you go. It's like a little Easter egg buried in the lecture. And then you have until Friday in class time to do this, and then I will let you know, yeah, what this is. It can be done, yeah. All right, so the alignment is then done using this mathematical technique called a hidden Markov model. And so we've linked to a separate video that discusses how hidden Markov models work. It's really neat. And in each state, you can either stay the same or you can change. And then the probability in the next state depends on what you did in the last state only. And so they use this for evolution. They'll take like the starting version of a thing and they'll take the ending, or rather they'll take two versions and they'll sort of walk these back until they connect, until there's a point where they are the same. And it's like, okay, how many steps to get from this common point to each of these? And it'll rerun that thousands and thousands of times, and it's really quick. It's a matter of seconds to do thousands of cycles of this, and you can get a really good alignment of profiles or of anything using hidden Markov models. And so the HHpred, the HH is for a hidden Markov model. That's the H. Yeah. And the HH is that it's hidden Markov model compared to another hidden Markov model. So they've abbreviated hidden Markov model, HMM, as just H. And so it's an HMM comparison, which is HH, that is used to predict. So that's HHpred. There you go. That's how the leopard got its spots. All right, so let's go back and look one more time at the HHpred method. So you submit something. HHpred goes out and does the legwork. It'll blast to get as many things that are confidently, statistically homologs as it can. It'll then align these using a hidden Markov model to generate position-specific gap penalties, like so. Once it's got that, it'll do another hidden Markov model to hidden Markov model alignment. So it's already made hidden Markov models for everything that exists, and it's comparing your thing to all the things in its database and telling you which, if any of those, it looks like and what the statistics are. Yeah. And then it spits out the results that are easy to read and take into account the sequence, the position of important, like conserved parts of the sequence versus not conserved parts, and the structure of your protein, or at least the predicted structure. It's really cool. All right, there you go, HHpred. Hope that helped.